Homologous recombination is the exchange of similar DNA sequences between different DNA molecules. Cells use homologous recombination to introduce genetic variation, as well as to repair damaged DNA. When you have completed this exercise, you should understand the double-strand break repair model of homologous recombination, understand how this mechanism differs from the Holliday model, and understand the cellular functions of homologous recombination. The mechanism of recombination in E. coli is called the REC BCD pathway. This mechanism closely follows the double-strand break repair model of homologous recombination. Therefore, the REC BCD pathway serves as a good model for recombination in both prokaryotes and eukaryotes, although the specific proteins involved differ from organism to organism. In the REC BCD pathway, homologous recombination is initiated by double-stranded breaks in a single DNA molecule, in contrast with the Holliday model, where recombination begins with single-stranded nicks at identical positions in each DNA duplex. In the double-strand break repair model of homologous recombination, the DNA is then processed to generate a gap in the DNA with a 3' single-stranded overhang or tail. In E. coli, the REC BCD enzyme processes blunt breaks in DNA molecules to generate such tails. REC BCD is composed of 3 protein subunits that are the products of the REC B, REC C, and REC D genes. REC BCD binds to the DNA at the site of the double-stranded break. REC BCD has both helicase and nuclease activities. It tracks along the DNA, unwinding the DNA, and frequently cleaving each strand. The cleaved DNA is destroyed. E. coli DNA contains relatively frequent 8-nucleotide crossover hotspot instigator sequences, commonly referred to as chi sites. When REC BCD encounters a chi site, its nuclease activity is altered. The mechanism of this change is unclear, but appears to be associated with either the inactivation or loss of the REC D subunit. Beyond the chi site, REC BCD no longer cleaves DNA from 3' to 5'. However, the 5' to 3' nuclease activity of REC BCD increases. This results in a 3' single-stranded tail on the DNA, terminating in the chi sequence. REC BCD helps to direct a protein called REC A to the single-stranded DNA tail. The active form of REC A is a protein DNA filament consisting of hundreds of REC A subunits, with 1 REC A for every 3 nucleotides of DNA. The filament has 2 distinct binding sites, a primary site with the single-stranded DNA molecule, and a secondary site. The REC A single-stranded DNA complex participates in a search for DNA homology. The secondary site binds a double-stranded DNA molecule. Binding in the secondary site is rapid, transient, and independent of DNA sequence, meaning the filament can quickly bind a double-stranded DNA molecule and scan along its sequence. Once a region of base pair complementarity is located, REC A promotes the formation of a stable complex between the single-stranded DNA tail in the primary site and the complementary strand of the double-stranded DNA molecule in the secondary site. As in the Holliday model, this step is called strand invasion. However, unlike the Holliday model, the REC BCD pathway can involve 2 Holliday junctions. Once the Holliday junctions have formed, REC A can dissociate from the DNA. The replication machinery then fills in the gaps in the DNA. This synthesis of new DNA is another feature missing in the Holliday model. The next step of the REC BCD pathway is branch migration. This step is similar to that of the Holliday model, except it can involve 2 Holliday junctions instead of just 1. In the REC BCD pathway, this step is catalyzed by 2 proteins, RUVA and RUVB. which form RUV-AB complexes at Holliday junctions. RUV-A protein is a DNA-binding protein that recognizes the gross structure of the Holliday junction, regardless of its specific DNA sequence. RUV-A recruits 2 hexamers of RUV-B to each Holliday junction to form RUV-AB complexes. RUV-B is an ATPase, and ATP provides the energy to drive the exchange of base pairs during branch migration. The final step of the REC BCD pathway is Holliday junction resolution. Again, this step is similar to that of the Holliday model, except it can involve 2 Holliday junctions. As will be shown, the possible products are the same: patch recombination products, also called non-crossover products, or splice recombination products, also called crossover products. In the REC BCD pathway, this step is catalyzed by the RUV-C endonuclease. For each Holliday junction, there are 2 possible cleavage sites. Site 1 consists of the 2 DNA strands that have not crossed over. Site 2 consists of the 2 DNA strands that have crossed over. If both junctions are cleaved in the same way, patch recombination products result. For example, if both junctions are cleaved at site 1, patch recombination products result. Patch recombination products also result if both junctions are cleaved at site 2. If the junctions are cleaved at different sites, splice recombination products result. For example, if the first junction is cleaved at site 1 and the second junction is cleaved at site 2, splice recombination products result. Splice recombination products also result if the first junction is cleaved at site 2 and the second junction is cleaved at site 1. Both prokaryotes and eukaryotes use the double-strand break repair pathway to exchange genetic information between DNA molecules, although the enzymes involved differ from organism to organism. This pathway can be used in several different cellular processes.